Good afternoon, or whatever this is. I realize I'm standing between you and lunch, and I never want to do that. So I'm going to take you on a fast ride. Hopefully they'll get my slides up. While I'm waiting for my slides, I'm going to take a little bit of a right turn. I've been presenting at this meeting for years, and I'm very proud to be able to do so. I've watched this group grow by an order of magnitude since I first attended. Being the third speaker in this session, where I realized that perhaps the two most laudable players in the space, besides, of course, my company, Celiad and Mesoblast, went before me, and I've watched the development of this field with great pride. And so I was going to end my talk with this quote, but I'm going to begin it. I've recently had the opportunity to read this book called The Emperor of All Maladies, which I highly recommend, talking about the development of cancer and then therapeutics for such. And this is a quote from Sidney Farber. The history of medicine is replete with examples of cures obtained years, decades, and even centuries before the mechanism of action was understood for these cures. So I'd like to begin with that and then take you on a journey as to why I think we're going to ultimately revolutionize the treatment of heart disease and ultimately other diseases as well. We're publicly traded, as you see. Shown here is our clinical pipeline. We have an allogeneic therapy, a cell therapy that is derived directly from the heart. We believe that our cells work as they do primarily because they are speaking the language of the heart. They come from the heart, they go back to the heart, and they trigger therapeutic regeneration, which we've shown previously in the clinical trial Caduceus. I've talked about that for several years now, where we showed a reduction in the amount of scar, an increase in viable, healthy living tissue in patients that had a big heart attack. From Caduceus, which was an autologous cell therapy trial, we moved to an allogeneic paradigm. And shown here, our product name is CAP-1002. We have 3 programs in various stages of the clinic where we are using this allogeneic cell. Our first trial is called HOPE. It's treating Duchenne muscular dystrophy. We treated patients with advanced heart failure, class 3 heart failure and dynamic. And then we've also just finished enrolling, as released today, the AllStar phase 2 clinical trial, which is the recapitulation of Caduceus with an indication expansion to 1 year post-MI and also using the allogeneic cells. I'm going to talk to you today primarily about our exosome program, where we've, again, worked hard to find the mechanism of action of the cells, and doing so discovered the exosomes and are using the exosomes to treat the eyes. I'd like to remind everybody that Janssen has an exclusive license option to CAP-1002. That's the cell therapy program for indications in the cardiology space. They paid us $12.5 million upfront, which has helped to power our research programs forward over the past several years. And also, most importantly, we have worked on a CMC, a manufacturing collaboration with Janssen over the past few years, which has given me enormous joy because we now have literally a commercial-ready product. So we went from a preclinical product in 2009 where we cut up biopsies into a dish and grew cells from it and treated 25 patients in Caduceus, to now a paradigm where we'll be able to develop 10,000 cells doses per heart to treat patients. And still in final stages of the development of this technology, and I think Fran is here from Janssen, who has worked on this with us, and it's been a really terrific collaboration. So really excited, and game on, J&J, because 6 months from now or thereabouts, you're going to get this data, and you have 60 days to decide whether to bring this therapy in-house for further commercial development and clinical development. So our cells, as I said, are developed from the heart. We use them to treat primarily heart disease. They have all kinds of biologic effects. We call them a local cell delivery or a local drug delivery system, all of these growth factors, and primarily our exosomes packaged into the cell. It is an off-the-shelf product. I was laughing a little bit earlier when we heard the talk about moving from a fresh to a frozen product. We have been able to do that. So our product is frozen and can be used up to 3 years. Our shelf life has been extended. And it has a 4-hour time from taking out of the freezer till it can be safely infused into a patient. So a lot of time to get it ready and get those patients infused appropriately. We have now infused over 100 patients. I don't know exactly the number in the clinical trials, but enough of them to assure that we have a very good safety record, primarily with the immunology of these cells, which we've been studying for a long time. And we have our intellectual property from our partners and also working to develop a full portfolio ourselves. So on this quick journey through cells, let me just tell you about AllStar. We announced this morning that the phase 2 of AllStar finished enrolling last week, which means that we'll be able to turn data over to J&J for their interim analysis in 6 months. It's powered for a reduction in the amount of scar 1 year post-treatment. And so just about a year from now, I'll be standing up here and sharing that data. I'm very excited about that. This has been going on for a long enough time that I am waiting anxiously for this data. We ended up enrolling 134 subjects and infusing such into 2 different cohorts, those that are 1 to 3 months post-MI. Those are the caduceus-like patients. Refer to our PNAS paper if you can't remember where we, what we did, and then also our Lancet paper where we actually published the clinical results of caduceus for the actual scar size reduction. We expanded that indication from 3 months to 1 year as an exploratory cohort to see if we could indication expand. And then we infuse the cells into the coronary arteries of the patients. Shown here, and I don't know why my graph isn't showing up. It's not meant to surprise you. It's just probably how the data came across. So I will just tell you that there is actually graphs that show that there is, in the phase 1 population, phase 1 clinical study, and the phase 2 equivalent population, a 50% reduction at scar size at 1 year. That's exactly what we're looking for in the phase 2, so a good roadmap for moving forward. And then we also saw improvements in ejection fraction. And if you'd like to see me afterwards, I can actually show you these graphs, so it'll be a good chance for us to get to know each other better. Dynamic was a very important exploratory study where we had some very important data in patients that have advanced heart failure. It was a phase 2A open label trial. It was a dose escalation study. In patients with advanced heart failure, you no longer just have one area of the heart that's affected by the heart attack, right? Like you would see in the AllStar or the caduceus patient population. So what we wanted to do is see if we could bathe the heart in cells and sort of mediate a global effect. And so we did up to 3 doses in each of the coronary arteries for a total dosing of 75 million cells. And these were, although we were targeting class 3 or ambulatory class 4, all the patients that came into Dynamic were class 3, and they were pretty sick folks. They had ejection fractions, which is the ability of the heart to meet the needs of the body, that was significantly attenuated and definitely impacting their ability to perform activities of daily living. And then we followed them for 6 and 12 months. And again, my graph is not showing up here, but what we saw is that significantly improved cardiac function, both structurally in terms of the volumes, which is very important, one of the only surrogate endpoints that's ever been approved for a therapy for heart disease, as well as improvements in ejection fraction. And that data was shown at 6 months at the American Heart Association last year. We'll be showing the 12-month data at the TCT meetings in the next few weeks. The beautiful thing about the dynamic data, which you can't see, so I'm just going to power right through it, is that there's a remarkable concordance of the data. We saw reductions in the structural remodeling issues in volumes. We saw improvement in contractility of the heart in terms of fractional shortening and end diastolic dimensions and end systolic dimensions. We saw improvement in the heart's ability to meet the needs of the body in terms of its contractile function and ejection fraction. And then we saw sort of the quality of life indicators, Minnesota Living with Heart Failure, 6-minute walk test, and the general picture was one of improvement in a patient population where the mortality rate is worse than most cancers. So a 5-year, 50% mortality rate is worse than most cancers at this point. And we now have something that shows that these patients that are in that miserable downward trajectory are improving. And we're very excited about the dynamic data. That's obviously going to take a large clinical trial to continue to validate those results, and looking forward to our partnership potentially with J&J to continue that work. So let me talk to you a little bit about the program, again, that's just finished enrolling that I feel really excited about, and that is the treatment of the cardiomyopathy associated with Duchenne muscular dystrophy. In this audience, it's funny, it's one of the few that I speak at where Duchenne muscular dystrophy is not one of the front and center topics because regenerative medicine hasn't really permeated into this space. And so Duchenne muscular dystrophy is when there's a mutation in the dystrophin gene. It means that the cell membranes can't respond to normal stresses. They get torn. Ultimately, it leads to cells dying mostly, starting in the skeletal muscle is where it's most visible. Boys are typically diagnosed 3 or 4 years of age when they start losing some of their milestones, and ultimately is a long, slow, steady decline of physical function in terms of their muscles until they die, typically in their early 20s. What most of the field did not pay attention to until very recently is that along with the skeletal muscle decline is cardiac muscle decline, and these kids get a very severe cardiomyopathy, which actually is the #1 cause of their death. And it wasn't until the parent lobby started to make real impacts into the field that the heart disease was focused on. So what we are, I don't know what's wrong with my slides. I've never actually had this happen before. So the previous data was our preclinical data, which showed that in the MDX mouse, which is the mouse model that is considered the gold standard for Duchenne muscular dystrophy, we showed an improvement in cardiac function with 1 dose of cells. We saw that we could repeat dose and re-synergize the effects. In fact, that effect is so strong that the parent lobby has come to us as HOPE has finished and said, can we now get another dose of cells? Is it possible to actually do a quick follow-on because we don't want to lose this response? We also saw an improvement in the mice ability to run on a treadmill. And that ability, as our cardiology thought leaders have told us, was disparate from what we saw as the improvement in ejection fraction. So we believe that there's also a synergy with the skeletal muscle benefit. And we have some data that was shown at the AHA last year suggesting a skeletal muscle benefit as well. So we took all that data. There's a lot more data. We can talk the mechanisms of that, but we took all that data and we did the HOPE Duchenne clinical trial. It was 25 boys, 13 got cells, 12 were standard of care controls. It means they were followed and got all the same measurements, but they didn't get an infusion. The 6-month data will be at the end of the first quarter of 2017. I'm really excited to see that data. This could make a big impact in the field, and especially with Sarepta getting approved on 12 boys with positive results in their trial. We're looking very, very carefully at the opportunities here to potentially register this product. So let me talk a little bit about our exosomes. Our exosomes are our new product. I started with a quote about not knowing mechanisms of action, but one of the beauties of Capricor is a deep academic bench behind us. And so we had these cells, and we've seen over and over and over that the data at 1 year is actually better than the data at 6 months. And how could that be with a paracrine effect? And so we went searching, and we actually discovered these exosomes. Exosomes have become the darling of the scientific community, although it hasn't quite permeated biotech. But what they are is they're nanometer-sized lipid bilayer vesicles packed with nucleic acids, primarily RNAs, and some proteins that actually are responsible for the mechanism of action of the cells. So we actually are honing in on what makes our cells work. So we did cardiology, cell therapy. We have a cell that treats the heart. Why on earth are you going after eye disease? Well, because what we realized when we discovered these exosomes is that they're so tiny, they're not yet well suited to really be circulating around, delivered, intravascularly. So we wanted to find a small compartment where a cell might not be indicated, and then we might want to be able to treat where the cells can actually marinate with each other and measure clinically the effects of these exosomes. And so we searched around, and we found that the best opportunity to use our cells with a high unmet medical need in the orphan disease space, which is all very much part of our business plan, was something called ocular graft versus host disease. And you just heard Donna talk about graft versus host disease, so I won't spend a lot of time on it, but I think everybody knows that it can be a side effect of any type of cell transplant that one might get. The eye disease that comes with it basically punctures holes into the epithelium, and patients report they feel like sand is being thrown into their faces, so very painful. This is the preclinical data. I'm going to leave you with this. We took this to the FDA in a pre-IND meeting this summer, and we are powering ahead towards getting into the clinic in the first part of 17 using our exosomes for ocular graft versus host disease. You don't need me to explain to you that the exosomes work better than the vehicle-treated control, and we are very excited at the opportunity to move forward with this therapeutic. And so, gosh, we're out of time, so I don't have time to talk about cash, but we just raised $10 million, so the bank account is full right now, and we're able to power forward and continue to put our head down and collect data and collect all of the very important information from the clinical trials that we have going on. And I'll leave you with this slide where we show where our upcoming milestones are. Jot them down, follow along with us at meetings, and stay tuned because I think it's going to be a very exciting year for Capricor in 2017. Thank you for your time today.